Building a Better World, CRISPR-Cas9 and Environmental Sustainability. Building a Better World, CRISPR-Cas9 and Environmental Sustainability. From enhancing crop resilience to aiding in wildlife conservation, CRISPR-Cas9 holds the potential to reshape our relationship with the environment and build a better, more sustainable world. Understanding CRISPR-Cas9, a brief overview. The system is inspired by the natural defense mechanisms of bacteria against viral infections. Essentially, it consists of two main components: the Cas9 protein, which acts as a molecular pair of scissors, and a guide RNA, which directs the Cas9 to the target DNA sequence. The versatility and precision of CRISPR-Cas9 have made it a game-changer in various fields, including medicine, agriculture, and now, environmental sustainability. Enhancing agricultural resilience. One of the most promising applications of CRISPR-Cas9 in the realm of environmental sustainability is its potential to enhance crop resilience. Climate change has brought about increasingly erratic weather patterns, resulting in droughts, heat waves, and new disease outbreaks that threaten global food security. Traditional breeding methods have limitations in developing crops that can withstand such challenges in a timely manner. CRISPR-Cas9 offers a solution by enabling scientists to introduce specific genetic modifications that enhance a plant's ability to tolerate stressors. For instance, researchers have used CRISPR-Cas9 to modify the genes of rice plants, making them more resistant to flooding. By altering the expression of certain genes involved in submergence response, scientists have been able to create rice varieties that can survive underwater for prolonged periods. This innovation has the potential to safeguard rice harvests in regions prone to flooding, ensuring a stable food supply for millions. Conservation and biodiversity. Biodiversity loss is another critical issue that threatens the delicate balance of ecosystems around the world. Habitat destruction, pollution, and climate change have led to the endangerment of countless species. Here, too, CRISPR-Cas9 offers a glimmer of hope. Scientists are exploring the possibility of using gene editing to aid in wildlife conservation efforts. One notable example is the use of CRISPR-Cas9 to combat the spread of mosquito-borne diseases. By modifying the genes of mosquitoes in ways that prevent them from carrying diseases like malaria or dengue fever, researchers aim to reduce the transmission of these diseases to humans. This approach not only protects human health but also contributes to the preservation of fragile ecosystems by disrupting the cycle of disease transmission. Mitigating pollution and waste. The impact of human activity on the environment is undeniable, with pollution and waste being significant contributors to environmental degradation. CRISPR-Cas9 technology offers potential solutions to mitigate these issues as well. Researchers are investigating the use of gene editing to create microorganisms that can break down pollutants and plastic waste. By harnessing the power of gene editing, scientists aim to accelerate the natural processes of biodegradation and waste breakdown, providing a novel approach to environmental cleanup. Ethical considerations and regulatory challenges. While the potential of CRISPR-Cas9 to contribute to environmental sustainability is undoubtedly exciting, it also raises important ethical and regulatory considerations. The ability to edit genes in a targeted manner brings both promise and concern. There is a need to strike a balance between the benefits of gene editing and the potential risks, including unintended consequences and unintended ecological disruptions. Furthermore, the international community must establish guidelines and regulations for the use of CRISPR-Cas9 in environmental applications. Ensuring responsible and transparent practices is crucial to prevent misuse and unintended negative impacts. The public's engagement and understanding of these technologies are also essential to foster informed discussions and decision-making. Conclusion. CRISPR-Cas9 represents a beacon of hope in the realm of environmental sustainability, offering innovative solutions to some of the most pressing challenges we face today. From enhancing crop resilience to aiding in wildlife conservation and pollution mitigation, the potential applications of this gene editing tool are vast and varied. However, as we venture into this new frontier, it is imperative that we do so with a strong commitment to ethical considerations, regulatory oversight, and open dialogue. Building a better world through CRISPR-Cas9 and environmental sustainability requires a collaborative effort among scientists, policymakers, ethicists, and the general public. Addressing challenges through research and collaboration. To fully realize the potential of CRISPR-Cas9 in the realm of environmental sustainability, ongoing research and collaboration across disciplines are essential. Scientists are working tirelessly to deepen our understanding of the ecological consequences of gene editing and to develop effective strategies that minimize risks. One area of research that holds immense promise is the creation of disease-resistant crops. As global food security becomes increasingly threatened by disease outbreaks that devastate agricultural yields, gene-edited crops with enhanced disease resistance could provide a solution. By carefully selecting genes that confer natural resistance to prevalent pathogens, researchers aim to develop crops that require fewer pesticides and have a reduced environmental impact. This approach not only safeguards the environment but also promotes healthier food production. Public engagement and education. Public acceptance and informed decision-making are crucial for the responsible development and deployment of gene-edited solutions. Educational initiatives can play a pivotal role in fostering a well-informed public discourse. By involving the public in these conversations, we can collectively shape the ethical frameworks and regulations that guide the responsible use of this technology. Regulatory frameworks for environmental gene editing. As gene editing technologies continue to evolve, regulatory frameworks must adapt to ensure the safe and responsible use of these tools. By implementing thorough risk assessments, regulators can make informed decisions about permitting or restricting certain gene-edited organisms or interventions. Balancing innovation and responsibility. The rapid progress of CRISPR-Cas9 technology presents both opportunities and challenges. While the potential benefits are undeniable, we must remain cautious about the unintended consequences that could arise from hasty or irresponsible gene editing practices. The delicate balance between innovation and responsibility requires a collaborative effort involving scientists, policymakers, ethicists, and the general public. An important aspect of this balance is maintaining transparency throughout the research and implementation process. Open communication about the goals, methods, and potential risks associated with gene editing projects is essential for building trust and ensuring accountability.